Now we know the various morphological changes that take place in a cell going through reversible cell injury. Up next, we have irreversible cell injury. See, till reversible cell injury, at any point when stress is removed, the cell is able to return back to its normal condition. But once irreversible cell injury takes place, even after the removal of injurious agent causing stress, the cell does not return back to its normal condition. That is why irreversible cell injury is in fact permanent cell injury. And that is why it is referred to as the point of no return. That is, once the cell is irreversibly injured, it will never return back to its normal original condition. Okay. Second thing, a cell that is irreversibly injured will eventually die. So cell death will take place. But the thing is that the pathways it follows to reach cell death are of different types. There are different pathways which the irreversibly injured cell can follow to eventually reach cell death. And depending on the pathway, the morphological changes that happen in the cell is different as well. Again, so there is a cell that is irreversibly injured. Now this irreversibly injured cell eventually dies. So there is cell death. It can go through different pathway to reach cell death. And there are two major pathways that it can follow. They are pathway number 1, necrosis, and pathway number 2, apoptosis. Both pathways, that is necrosis and apoptosis, will eventually result in the death of the irreversibly injured cell. But the difference lies in the morphological changes that the cell goes through as it follows the pathway. That is, when the irreversibly injured cell follows pathway number 1 or the pathway of necrosis, the morphological changes are different as compared to it following pathway number 2. And that is why in case of reversible cell injury, we simply learnt about the various morphological changes that take place in reversible cell injury. But in irreversible cell injury, it's best to learn the morphological changes taking place in the cell on the basis of the pathway that it follows. Starting off with the very first pathway, that is the pathway of necrosis. So, what is necrosis? Necrosis refers to the series of changes that occur in an irreversibly injured cell as it eventually reaches cell death. Okay, there is irreversibly injured cell. The injury goes on becoming worse and worse and worse, and the cell ultimately dies. As the situation becomes worse and worse and worse, the cell goes through a series of morphological changes. And this series of morphological changes that take place in an irreversibly injured cell leading ultimately to its death is a necrosis. But this can even be apoptosis. Because I said earlier, the series of changes is different for different pathway. This series of changes could be for necrosis, or this series of changes could be for apoptosis. Because in both the conditions, the cell starts with irreversible cell injury and ends with cell death and goes through a series of changes. So is it necrosis or apoptosis? So it is necrosis when this series of changes is due to 4 major characteristic features. Characteristic features. When it is due to these 4 characteristic features, 1, 2, 3, 4. Only then can we call that particular series of changes to be necrosis. Okay, one last time. What is necrosis? It is the series of changes taking place in irreversibly injured cell as it reaches cell death, where this series of changes taking place is due to 4 major characteristic features. They are these. And let me explain you each one of them. First, denaturation of the protein. When the cell gets injured, Irreversibly, and it goes through necrosis, the protein of the cell gets denatured or destroyed. And there are two types of protein: structural protein and functional protein. The structural protein is responsible for maintaining the structure and integrity of the cell, and the functional protein includes enzymes that perform various functions. So if structural protein gets destroyed, there is loss of structural integrity of the cell. And when functional protein gets destroyed, there is loss of function of the cell. So what takes place? Denaturation of structural and functional protein within the cell takes place. Number 2, leakage of cellular content. See, normally the cell with its plasma membrane is intact. In reversible cell injury as well, the plasma membrane was intact. But in necrosis, the plasma membrane disruption takes place. The plasma membrane disruption takes place. So if the plasma membrane gets disrupted and there is gap in between plasma membrane, things from inside the cell will leak outside. So there is leakage of intracellular content. Leakage of intracellular content. There is disruption of plasma membrane that leads to leakage of intracellular content. When this intracellular content from inside the cell leaks outside, it contains what? It contains enzymes, cellular components that irritates the surrounding. Irritates the surrounding. And the surrounding contains blood vessel. And when blood vessel gets irritated or injured, it shows vascular response. What is the response that vascular tissue shows when it is injured? Inflammation. So there is in fact local inflammation. So remember, necrosis is always associated with inflammation. That is what I am referring to. In pathway number 1, the morphological changes are different as compared to pathway number 2. In necrosis, there is inflammation. In apoptosis, there is no inflammation. In necrosis, there is disruption of the plasma membrane. In apoptosis, the plasma membrane remains intact. In necrosis, a mass of cell, one cell, the surrounding cells, a mass of cell goes through necrosis at once. In apoptosis, few cells or even one singular cell might just go through apoptosis. Necrosis is always pathological, that is seen in diseased condition, but apoptosis could be physiological or pathological. How? That I'll tell you under the topic of apoptosis itself. So wait for that. Okay, so what has taken place? Local inflammation has taken place. As a result, inflammatory cells like neutrophils come out. Now, the cause of the inflammation could be the leakage of the intracellular content or a bacteria or a microorganism. If the necrosis is due to microorganism, what is necrosis? It is cell injury. Cell injury is caused by an injurious agent. If in this case the injurious agent is microorganism, then the cause of inflammation could be microorganism. But if it is not microorganism, then intracellular content coming outside can irritate the vascular tissue, leading to local inflammation. Okay, the concept is clear. Okay. Next, enzymatic damage. See, due to necrosis, due to damage to the cell, the lysosomes within the cell, they burst, and the lysosomal enzymes from inside comes out into the cell, and it starts eating the cell from inside. The self-eating of the cell by the lysosomal enzyme is referred to as autolysis. Second, the inflammatory cell like neutrophil, they eat the cell from outside. This is heterolysis. So what is autolysis? That is self-eating of the cell by lysosomal enzymes. What is heterolysis? That is eating of the cell from outside by WBCs. So autolysis, cell gets eaten from inside by lysosomal enzyme. Heterolysis, cell gets eaten from outside by neutrophils or macrophages. So that's necrosis. This is actually necrosis. This whole thing defines the word necrosis. What is necrosis? Necrosis refers to the series of morphological changes taking place in an irreversibly injured cell that eventually results in the death of the cell. The series of morphological changes is as a result of four characteristic features. Number one, denaturation of the protein, where there is loss, where there is degradation or destruction of both structural and functional protein. Structural protein is responsible for providing structural integrity. Functional protein includes enzymes that carry out various activities. Number two, there is leakage of intracellular content due to disruption of plasma membrane that triggers local inflammation, and there is enzymatic digestion either due to due to lysosomal enzyme from inside and WBC from outside. That's necrosis. Okay. What is the clinical significance of leakage of cellular content outside? It has clinical significance as well. See, here's the clinical significance. This cell right here is going through the process of necrosis. That is why there is disruption of the plasma membrane. Due to disruption of the plasma membrane, the intracellular content, it leaks outside. Some of these intracellular chemicals, as I'm showing in green, is specific to the cell. That is, it is specific, just found in this cell. So these intracellular content, they leak out as well, and then enter where? They enter into the blood circulation. So when you measure their serum level, they are found to be elevated. The level of these chemicals found within the cell. See, these chemicals are present within this particular cell. But now, since there is disintegration of the plasma membrane, they have leaked into the blood circulation, and their level has risen in the blood circulation. There is rise in their level in the blood circulation. So when blood test is carried out, the serum level of that enzyme, the serum level of that chemical is found elevated. And that is used as a marker for diagnosis of this cell injury or necrosis of the cell. So you are diagnosing the injury to the cell with the help of the leakage of this cell specific chemical into the blood. And you carried out blood test for that. And this particular chemical used right here is referred to as biomarker. Biomarker. So what are biomarkers? They are biochemical markers that allow for the diagnosis of injury to a specific cell. Why specific cell? Because the chemical, the biomarker, is also usually specific to that cell. Let's look at the biomarkers of various important organs or important cells in your body. First, the heart cell. Just remember, my time to call. Y for myoglobin, T for troponin, another T for troponin, C for CKMB, A for AST, and L for LDH. Out of all these markers, the most important one is troponin I. See, what is the significance of troponin I as a biomarker anyway? Or what is the significance of biomarker? I said that when the necrosis takes place, here the necrosis of a heart cell is taking place. Let's say the patient has heart attack due to a heart attack, and at that time the necrosis of the heart cell is taking place. Okay. So when necrosis of the heart cell takes place, the cell goes through morphological changes. But even before these morphological changes are appreciated under light microscope, again I repeat, even before these morphological changes of the heart cell are appreciated under light microscopy, there is already the presence of biomarker in the blood. So this is faster than the morphological changes of light microscopic morphological changes. What is it? It is faster than light microscopic morphological changes. That's the significance. Okay. The second is hepatocyte or liver cell. So it has transaminases, aspartate transaminase, alanine transaminase. Another is bile duct epithelial cell. So it has alkaline phosphatase isoforms. That's it. Okay, so I said that these are the characteristic features, the reasons behind the series of morphological changes that take place in the cell during necrosis. The question is, what are these changes then? So these morphological changes, I'm going to divide it into two. One that is appreciated under electron microscope, and the other that is appreciated under light microscope. First, electron microscope. Under electron microscope, you are able to appreciate the following. I'm just going to draw a normal cell with normal nucleus, and then it has a mitochondria at the side. Now this normal cell goes through the process of necrosis. As it goes through the process of necrosis, there are morphological changes taking place at 3 places, which is appreciated under light microscope. Number 1, changes at the level of plasma membrane. Number 2, changes at the level of cytoplasm. And number 3, changes at the level of mitochondria. So we have number 1, number 2, and number 3. Number 1 at the level of plasma membrane. Number 2 at the level of cytoplasm, and number 3 at the level of mitochondria. First, at the level of plasma membrane. What's happening to the plasma membrane? Disruption of the plasma membrane takes place. As a result, the content from inside leaks outside. So there is disruption of plasma membrane. So when plasma membrane gets disrupted, the content from inside leaks outside. But things from outside can also go inside. What is outside? Lots of calcium is present outside as compared to inside. All these calcium can now leak inside the cell, resulting in elevated level of intracellular calcium. It's boring without a diagram, so let's look at the diagram. This is the cell going through necrosis. You can see that the plasma membrane is disrupted. As a result, the calcium from outside can easily leak inside. And when calcium from outside leaks inside, there is increase in the level of intracellular calcium. And the thing is, calcium is notoriously known for activating various intracellular enzymes. So in this case as well, it is going to activate 3 major enzymes, namely phospholipase, protease, and nuclease. Really easy to remember. Just look at a typical cell. So first is an enzyme that disrupts the plasma membrane. Second is the enzyme that disrupts the protein within the cytoplasm. And third is the enzyme that disrupts the nucleus. 1, 2, 3. First, phospholipase. It has A's at the end, so obviously it's an enzyme. What kind of enzyme? The enzyme that destroys phospholipid. And where is phospholipid present? It is present in the membrane of membrane-bound organelle. See, right now I'm talking about the membrane of membrane-bound organelle and not the plasma membrane. Remember that I'm talking about membrane present within the cell. Okay. So what it does is that it breaks down the phospholipid. Breaks phospholipid of the membrane of membrane-bound organelle. And the phospholipid, broken down phospholipid, it curls within itself. It curls within itself. What does phospholipid contain? It contains phosphate. Once the phospholipid is broken down, the phosphate now becomes free. And the phosphate is negatively charged. This negatively charged phosphate, it attracts calcium. So what you get is broken down phospholipid plus little amount of calcium present in whorled pattern or concentric lamellated curl. This is referred to as myelin figure. And if you remember correctly, I have already mentioned myelin figure under the topic of reversible cell injury, which suggests that myelin figures are present both in reversible and irreversible cell injury, but obviously it is present in way high amount in irreversible cell injury. Second, protease, which breaks protein. And then third, nuclease, it breaks nucleus and brings about nuclear changes. What are the nuclear changes? I'll explain soon enough. Okay, so that's first. Second, at the level of cytoplasm. I said that during necrosis, denaturation of the protein takes place, and these denatured proteins are present in the form of fluffy aggregates. Fluffy aggregates within the cytoplasm. Here are the fluffy aggregates of the protein. Plus, myelin figures are there. Where did myelin figures come from? From the disruption of the plasma phospholipid present in the membrane of membrane-bound organelle. So there are myelin figures within the cell. myelin figure, sorry, green. Myelin figures within the cell. I'm representing it with the color green. Okay. Third, mitochondria. So the mitochondria swells up, plus it has amorphous, flocculent densities. What are they? So the calcium, they enter inside the mitochondria. Inside the mitochondria, the calcium, they deposit in the form of amorphous bodies, and these amorphous bodies present within the mitochondria are the amorphous flocculent densities of mitochondria. So those were the three changes at three levels that are appreciated under electron microscopy when a cell is going through necrosis. Next, we have electron microscopy. So let's look at the morphological changes that is appreciated under electron microscopy. I have divided it into two: cytoplasmic changes and nuclear changes. So first, let's look at the cytoplasmic changes. Here is a normal cell. Okay, this is a normal cell. It is not going through necrosis. It's a normal cell. The cell is under H&E stain. H&E stain. H stands for hematoxylin, and E is for eosin. Hematoxylin gives blue stain and eosin gives pink stain. Hematoxylin positive is nucleus, and all the dots that you see here, they are ribosomes. So nucleus and ribosomes, they give stain to hematoxylin. Eosin, on the other hand, is for cytoplasm. So the complete cytoplasm is stained pink due to eosin. Okay. Now this normal cell goes through necrosis. What are you able to see? What kind of changes are you able to see? You see two major changes first. Number one, increase in eosinophilia. That is, the cell becomes more pink. And number two, glassy homogeneous appearance. Glassy homogeneous appearance. That is, if you look at the cytoplasm, it is homogeneous, clear. The reason behind increase in eosinophilia is two. There are two major reasons behind it. Number one, denaturation. Denaturated protein take more eosin. What do I mean by this? See, the reason the cytoplasm of the cell is eosinophilic is due to protein. The protein within the cytoplasm of the cell takes eosin and imparts pink coloration. When this protein denatures, it opens up, which increases the surface area. Now to this increased surface area, even more eosin goes and sticks. As a result, the cytoplasm becomes even more eosinophilic. So there is increase in eosinophilia. Second reason, loss of basophilic ribosomes. Ribosomes, as you saw in the normal cell, they are blue in color because they take hematoxylin stain, right? Due to necrosis, there is loss of ribosomes. So if ribosomes are no longer there, there is decrease in basophilia, decrease in blue color, or eventually increase in pink color. Glassy homogeneous appearance due to loss of glycogen granules. See, the cell contains stores of glucose referred to as glycogen granules. When it is irreversibly injured, the glycogen granules containing glucose gets used up in order to produce energy. So all the glycogen granules, they get used up. So there is loss of glycogen granules. When the granular structure is lost, the background becomes plain and smooth. That's why glassy homogeneous appearance. Okay. Then I said that the lysosomes present within the cell, they burst and release lysosomal enzymes. These lysosomal enzymes, they destroy the cellular organelles. And at these areas with cellular organelle, now there are no longer cellular organelle, so they appear empty. See, these empty spaces, they had cellular organelle, but now the cellular organelle have been digested. So there are empty spaces that give moth eaten appearance. Why? Due to destruction or dissolution, due to dissolution of cell organelle. Okay. During necrosis, there is disruption of the plasma membrane, and eventually the complete cell gets destroyed and the plasma membrane containing phospholipid gets destroyed as well. When phospholipid gets destroyed, it curls within itself. The phospholipid, it curls up. The phospholipid curls up. This curl of phospholipid is referred to as myelin figure. Again, the myelin figure that I was talking about during electron microscopy were the myelin figures that are present within the cell. And these myelin figures were obtained from phospholipid of membrane-bound organelle. But this large myelin figure right here is obtained from the membrane of the cell itself, from the phospholipid of the membrane of the cell itself. Previously, under electron microscopy, these myelin figures were obtained from cell organelle. This myelin figure is obtained from the cell itself. So it is much larger. But still, it contains phospholipid. And when phospholipid gets broken down, what happens? Phosphate ions are released. They are negatively charged. They attract calcium ions. So calcium ions go and stick to this myelin figure. The same thing is happening here as well. Now, what do we call when calcification of dead cell takes place? The calcification of dead cell is referred to as dystrophic calcification. So here dystrophic calcification has taken place. First, you have the formation of myelin figure, and this myelin figure has two fates. Either it gets phagocytosed, that is, it gets eaten up by WBC, or it goes through dystrophic calcification, that is, calcium goes and sticks on it. Dystrophic calcification. These are all the morphological changes, the cytoplasmic changes. As for nuclear changes, here we are. This is a normal cell with a normal nucleus. Now the rest are the cell going through the process of necrosis, and the nucleus are different as well. In first case, you can see that nucleus is condensed. This is referred to as pycnosis. Pycnosis is nuclear condensation or nuclear shrinkage. As a result, what you get is a pinpoint nucleus. Next is karyorexis. Karyo refers to nucleus and rexis refers to fragmentation. So there is fragmentation of the nucleus. Next is karyolysis. Karyo refers to, again, nucleus, and lysis refers to dissolution. So these are the various morphological forms of nucleus that are present during necrosis. During necrosis, you get to see any one of them. You can either get to see pycnosis, where there is condensation of the nucleus, or you get to see karyorexis, where there is fragmentation of the nucleus, or you get to see karyolysis, where there is dissolution of the nucleus. Be it pycnosis, karyorexis, or karyolysis, eventually the nucleus gets completely destroyed, either through this pathway or this pathway or this pathway. And with that, we are done with the morphological changes seen in an irreversibly injured cell when it follows the pathway of necrosis. We saw the morphological changes both in light microscopy and electron microscopy, and we saw the 4 characteristic features that were the cause behind all these morphological changes. You won't find any better conceptual video for the topic of necrosis morphological changes other than this. As for notes, all notes are available in PDF form for free in my Telegram group, for which the link I have in the description or in the comment section.